Hello everyone, my name is Jiayi. Today I'm very happy to share with you this interesting topic, Interpretable Machine Learning for Genomics. In Lecture 18, Professor Luo has taught us about the difference between AI and machine learning, as well as showed us some examples for AI in genomics. We all know that machine learning is driving great progresses in this genomic big data era. Just a quick recap, machine learning is a subset of AI technology that gives computers the ability to learn without being explicitly programmed. Machine learning models have the ability to identify complex patterns in data, so they are gaining increasing popularity in genomics. However, there is a problem that machine learning algorithms are facing. That is the trade-off between interpretability and accuracy. First, let's define interpretability. A widely accepted definition is a degree to which a human can consistently predict the model's result. Here is a graphical illustration of the interpretability-accuracy trade-off. The orange dots are all machine learning algorithms. And as you can see, when the model becomes increasingly complex, while the accuracy goes up, the interpretability goes down. You might want to ask, why does that matter? Well, a dramatic scenario is captured in this movie, where a near-human intelligent AI called Hal was sent to the outer space with some scientists. However, Hal decided to kill these scientists. But these scientists has no idea why. I bet the scientists wish they had pre-installed some explainable AI module. Although our AI algorithms nowadays still has a long way to go before reaching AGI level, model interpretability is still of vital importance for research. So, why machine learning model explainability is important? The first reason is that it can help us troubleshoot. Troubleshoot is to identify the mistake or biases in data or issues with how the model is trained. Here is an example. In these scenarios, the researchers found their image classifier consistently misclassify husky as wolf. After using some IML method, they found that the model is actually paying a lot of attention on the snowy background. This is how they go back and examine the data and found that all the wolf image appear in a snowy background. Hence, by applying IML method, they know that they need to debias the data. Apart from the troubleshooting, IML method can help to enhance our scientific understanding. The reason why we want to apply those complex ML method is that they can extract biological insight from large-scale data that the previous method could not achieve. However, it could not add on our scientific knowledge if we don't know what the model learned. I really like this graphical illustration here. There are 5 layers in this framework, and interpretability method is like an additional layer on top of the black box model to extract human intelligible knowledge. Last but not least, it's about trust and acceptance. IML method can help us understand what is the underlying decision-making process of the black box ML models so that it can enhance acceptance among researchers, clinicians, and relevant stakeholders. After convincing you IML is important, let's take a look at some readily available IML tools. The first resources that I want to point you to is this wonderful IML textbook authored by Christopher Molnar. This book is open source on GitHub, and I put the link here. You can always refer to this book if you want to know more further detail about IML methods. Now let's take a look at the IML taxonomy. There are actually a lot of IML methods being developed to serve different purposes, and different IML methods tell a different story. By having an understanding of the taxonomy, you will know what is a suitable method for your problem. So firstly, IML method can be classified whether they are local method or global method. Local method only offers the explanation for a single prediction, while the global method tells you how the input variable is linked to the overall result. Also, there are IML methods developed to cater for specific data type, such as tabular data, text data, image data, and graph data. This interpretability method can also be classified as for what purposes they serve. Whether they are intrinsically interpretable methods, that is to say, these machine learning models are white box. You can look at it and know what do they believe are the most important features. On the other hand, there are post hoc methods, where you already have a trained black box model, and you want to use IML methods to give you explanation for these complex models. There are also IML methods served for other purposes, such as enhancing the fairness of model or test the sensitivity of prediction. Last but not least, IML methods can also be classified whether they are model-specific or model-agnostic. Model-specific method can only be applied to a specific class of model, whereas model-agnostic method can be used to explain all sorts of machine learning models. After understanding the IML taxonomy, it is also beneficial for us to know what is the underlying implementation of this IML method. There are 3 general IML strategies. The first strategy is called probing. Probing inspects the inner logic of a trained model. It can tell you how the model believes is the importance of each feature as well as the interaction of different features. The second strategy is called perturbing. Different with probing, which inspects a model, perturbing manipulates the data. For example, you can remove some features from the dataset and see how the model prediction is changed. Perturbing is usually used to answer questions related to sensitivity, or it can be used to answer counterfactual questions, such as what if feature X is of a different value. The last strategy is called surrogate. Surrogate method train a much simpler model called surrogate model to approximate the prediction of the complex black box model. By looking at the explanation of the surrogate model, you can explain how the black box model makes a prediction. A gentle reminder, whenever you encounter some problem and you believe that you need some interpretability method, the first thing you want to do is to search online before you reinvent the wheel. Now, let's take a look at a real-life research which apply IML in genomics. We will have a look at this research paper titled MethodNet. Basically, in this paper, researchers use IML to extract biological insight from DNA methylation data. DNA methylation sounds familiar, right? Because we already learned it in the lecture. Just a quick recap, DNA methylation, or DNA-M for short, is the addition of a methyl group to a nucleotide. DNA-M does not alter the DNA sequence and occur most frequently to cytosine guanine dinucleotides, or CpG for short. DNA-M near gene promoter can suppress transcription, and DNA methylation data analysis is used for epigenome-wide association studies, or EWAS for short. What EWAS does is that they test association of DNA-M with phenotype, exposure, and state of human health and disease. 2 additional pieces of background information before we proceed. Actually, DNA-M can be used to estimate chronological age, and the estimation is called DNA methylation age. Some example of such estimator is the Hannum clock. Another term, biological age, also called epigenetic age acceleration, is the difference between chronological age and methylation age. For example, a 40-year-old alcoholic person who has a methylation age of 50 years is said to have an age acceleration of plus 10 years. Biological age is often associated with disease risk. However, the problem with DNA-M-based age estimator is that they offer very limited interpretation of how CpGs influence biological age. Thus, the researchers want to apply IML to decode the interaction of CpGs underlying the aging process. Remember our 5-layer interpretation framework? We can use this to help us understand the framework proposed in the Mesonet. So for the world layer, CpGs can influence an individual's biological age. And this was captured in our data, which consists of the methylation data of individual as well as their corresponding biological age. And the black box model is applied to analyze this data. The underlying architecture of the black box model is a variational autoencoder together with a multi-layer perceptron to make the final prediction. And the explainable machine learning method used is called SHAP, S-H-A-P. Let's have a quick introduction of SHAP score. SHAP is a model-agnostic IML using probing strategy. It has a solid theoretical foundation in game theory. A positive SHAP score indicates that the feature can positively impact the prediction and vice versa. The magnitude of the SHAP score measures how strong the effect is. Here are some results from the study. The researchers rank all the CpG features according to their SHAP score, and found that the top 1,000 CpG features that Mesonet believe is the most important corresponds to the well-established Hannum clock. In addition, they were able to find the correlation among SHAP contribution so that they can identify the common important CpGs across different age groups. In this way, IML helps researchers uncover the defining CpGs for the aging process. The researchers also conducted some external validation to make predictions of smoking status on another dataset and compare it to prior EWAS meta-analysis. In the graph below, you can see that the ranking according to SHAP score corresponds to the gold standard EWAS ranking. So we have seen that AI for genomics has huge potential, and whenever you encounter such problem, don't forget our 5-layer interpretability framework. To conclude this presentation, I would like to offer some future prospects. To summarize, we have seen that machine learning model interpretability is important, and many IML methods has been developed to serve different purposes. Also, applying IML to genomics can offer us new insights. As for outlook, we still need to develop novel IML method for heterogeneous and high-dimensional data. Additionally, we need to benchmark current IML method for different genomic applications. Last but not least, it is important to integrate domain knowledge of complex biological systems so that we can develop better IML methods for AI in genomics. In the end of the presentation, I would say special thanks for Professor Luo, TAs, and all my wonderful classmates. Thank you so much.